Welcome. Today we are going to discuss about the muscular tissue. As we all know that tissue means the cells working together in functionally related groups. That is called tissue. And types of tissue we already discussed about the epithelial tissue, then connective tissue, which is helpful in support, and then muscular tissue, which we are going to discuss today, which is responsible for the movement, and then nervous tissue, which is responsible to control all the organs. Then muscular tissue, that is the skeletal muscle tissue. Then we have cardiac muscle tissue, and then smooth muscle tissue. So first is the skeletal muscle tissue. And what are the characteristics? They are long and cylindrical cells. They are multinucleated, means the cells are multinucleated and having the striations because of presence of actin and myosin proteins. So we can see here the nucleus. They are multinucleated, means a single myofibril is having more than one nucleus, and the part of muscle fiber is visible like this. Then function, it is responsible for the voluntary movement. Whatever the movement of the body which is voluntary under control, then they are responsible. And then we can see the manipulation of the environment, facial expression, as well as then we can see the location that the skeletal muscle attached to the bone and occasionally to the skin. Then composition, a muscle, that is the fibril structure. Then it is a number of myofibrils, means a number of fine myofibrils are enclosed in a connective tissue sheath called endomysium and form the muscle fiber. Okay, so myofibrils, that is the numerous, they are enclosed in the connective tissue covering, that is called endomysium, and the structure then is called muscle fiber. Then many muscle fiber are enclosed in another connective tissue membrane or septa, that is known as perimysium, and form the muscle bundle of fasciculus, that is also called fasciculus. Then a good number of fasciculi are enclosed or covered outside by connective tissue coat, that is called epimysium, and the muscle is formed. Then muscle fiber are considered to be the unit of the muscle. Then come to the structure. So muscle fiber are composed of elongated tubular cells with multiple nuclei and extended along the muscle with a length of about 1 to 40 millimeter and 10 micron to 100 micron in breadth. Then sarcolemma, outer transparent cell membrane of the muscle fiber, like other cell membrane, it is composed of outer and inner protein layers and an intermediate lipid layer. Now we can see here, this is how we can see they are enclosed. So these each are the myofibrils, and the myofibrils are enclosed in a connective tissue sheath that is called endomysium. So like this we can see the numerous bundles are there. They are forming the structure that is called muscle fiber. Now here also we can see the muscle spindle in the transverse section. It appears like this. So in this photograph we can see that these are the myofibrils. 1 2 3 4 5 6 7 8 9 myofibrils are visible. They are enclosed in a covering that is called endomysium, and we can also see that the striations are visible, and nucleus are multi. We can see that one or two are visible here. Then mitochondria are also there, and sarcoplasmic reticulum is also there, sarcoplasm is there. So like a cell there is in the form of a structure that is similar to the cell, and the plasma lemma here it is called sarcolemma. Endoplasmic reticulum is here called sarcoplasmic reticulum. Okay, so and cytoplasm is called sarcoplasm here in this kind of cells, where we are having the numerous myofibrils which is enclosed in a covering called endomysium. Now the sarcolemma maintains the higher concentration of sodium and chloride outside. So sarcolemma of the muscle fiber, in this way they are maintaining the concentration of ions and higher concentration of potassium inside, which results in potential difference, that is of -70 millivolt between them. And with the arrival of the nerve impulse at the motor end plate, what is motor end plate? The junction of the nerve endings with the muscle fiber. So when the arrival of the nerve impulse at the motor end plate, the potential difference abolishes and depolarization occurs, and the muscle fiber contract, And inside sarcolemma, elongated myofibrils, multiple nuclei, mitochondria, small Golgi apparatus are embedded in a sarcoplasm. Then sarcoplasm, it is semi-fluid, non-contractile cytoplasm of the cell, muscle cell, where other constituents of the cell are embedded. And then nuclei. So in this case, numerous elongated nuclei are found beneath the sarcolemma, and several hundred nuclei may be present in a single muscle fiber. Then mitochondria or sarcosomes are situated in row in between the myofibrils and provide energy to the muscle. Then myofibrils. These are parallel thread-like structures situated along the length of the muscle fiber. And each myofibril is composed of longitudinally arranged myofilament, that is fine myofilament, and myofibril present along its length alternate dark band and light bands, and the dark band is roughly refractive and anisotropic in nature because of overlapping of actin and myosin. Actin is thin filament, myosin is thick filament, and that is also called A band. Then light band is non-refractive and isotropic in nature, and is called the I band. That is the we can say that isotropic type. Then these A band and I band of adjacent myofibrils are aligned transversely and give a muscle fiber a cross-striated appearance, as we can see in this photograph here. In this way, this is the zone of the A band, this is the zone of the A band, and in the mid this is the zone of the I band. So why this is called A band? Because of they are anisotropic. Anisotropic means because of overlapping of myosin, that is thick filament, that is appearing here pink, and violet is the thin actin filament, and overlapping is occurring during contraction. So this is why this is called anisotropic. We can see in another photograph also. So this is the showing the here A band because of overlapping is occurring here, and in this way we can find here the I band. Now we can see that at the middle of the I band, a thin dark transverse line is present known as Z line or Z disc, and the portion of the myofibril in between two adjacent Z discs is known as sarcomere, and the sarcomere are considered to be contractile unit of the myofibril. Then the A band is bisected at the middle by a lighter zone known as H band, and within the H band a narrow dark line is called M band. Then myofilaments are the fine thread-like protein filament arranged longitudinally within the myofibril, and they are the three varieties: myosin, actin, and tropomyosin. Then myosin filaments are thicker and situated longitudinally parallel to each other in the dark A band, and these filaments become more thick at the middle during contraction. Next point about the actin filament are thin filament and long and extend from the Z band towards the A band, as we can see here. This way the actin filaments is there. So this is the Z line. In the mid, this actin filament is extending on both sides like this. Then tropomyosin protein filament originate at the groove between the two strands of the helical action protein or action filament or actin filament. Next is the sarcoplasmic reticulum. They are the myofibrils are surrounded by smooth surface circular and branching tubules derived as tubular ingrowth from the sarcolemma, and these tubules are called sarcoplasmic reticulum, and the circular tubules are known as centrotubules and extend the Z disc and also called T system membrane. So we can see in this way these structures are found. In this electron microscopic photograph also we can see. So this is how we can see the Z disc on both the sides. This is the M line. Okay, so this is the in the and where overlapping is found, this is the A band, and this is the I band. Here not overlapping is there. Next point, in this photograph we can see the muscle cell. This is the muscle cell. Actually here we can see the difference. This is the spindle shaped cell. They are smooth muscle cell found in the urinary bladder of pig. So this is the difference. There is no striations in the smooth muscle cell, but skeletal muscles we can find the difference, that is the having the striations. We can see here these are the striations are visible. So we can see the muscle cell is fifth one, and sixth is the nucleus of the muscle cell, which is multinucleated. We can see the numerous nuclei are visible, So this is the skeletal muscle of the tongue, and we can see along with that the perimysium is visible like this, and these are the muscle fiber which is enclosed by the endomysium. So and the muscle fiber is having numerous myofibrils like this. Now in this photograph, very clearly observed, we can see that the striations are very clearly visible. So we can say that these are the striations. In this way, the lines like structure as they are the protein. These are the nuclei which are multinucleated, we can see. Now in this photograph also, this is a transverse section, it appears like this, and this is the longitudinal section. In the longitudinal section, striations are more clear, but in the transverse section we can see in the form of bundles. Now here in this photograph, motor nerve plexus, and because nerves are coming in this way, we can see the nerves and the muscle fiber, they are motor end plate visible here. Isotropic and anisotropic bandings are also, that is striations visible. Endomysium with fibroside nucleus is also there. So this is the skeletal muscle fiber of the cutaneous muscle in cat. This is the electron microscopic photograph showing you the myofibrils, T system, sarcoplasmic reticulum, mitochondria, and scanning electron microscopic photograph is appearing like this. We can see the heterochromatin, euchromatin inside the nucleus, which is having the chromatin material, and collagen fiber endomysium is also there, and we can see the Z line A band appear in the electron microscopic photographs. Now cardiac muscle fiber or cardiac muscular tissue, they contract to propel the blood into the circulatory system, and characteristic of these cells, they are branching cells. So we can see that they are branched in like this. So in this way we can see the branching is appearing. Next point, we can see in this photograph that the are uninucleate, they are intercalated disc. We can see that intercalated disc are visible, that is the cell junction, and then location where we can find occur in the wall of the heart. And cardiac muscle is striated muscle that is present only in the heart, and cardiac muscle fiber have a single nucleus and are branched and joined to one another by intercalated disc that contain gap junction for depolarization between the cells and desmosomes to hold the fiber together when the heart contract. So here we can see, if you cut the wall and appearing like this, parietal pericardium, visceral pericardium, then pericardial cavity in between them, then myocardium, and then endocardium. Next we can see that the cardiac muscle tissue is we can see like this. This is the cardiac muscle tissue in which we can see the branching, the striations are visible, nuclei are many, that is visible here. Now come to the T tubule. T tubule are microscopic tube that run from the cell surface to deep within the cell, and they are continuous with the cell membrane and are composed of some phospholipid bilayer, and are open at the cell surface to the extracellular fluid that surround the cell. So in this way these tubules are opening outside. Next is the T tubules in the cardiac muscle are bigger and wider than those in the skeletal muscle, but fewer in number. In the center of the cell, they join together, running into the along the cell as a transverse axial network. Inside the cell, they lie close to the cell internal calcium store and sarcoplasmic reticulum. Here a single tubule pairs with the part of the sarcoplasmic reticulum called the terminal cisterna, in combination known as diad. The function of the T tubule include rapidly transmitting electrical impulse known as action potential from the cell surface to the cell core, and helping to regulate the concentration of calcium within the cell in a process known as excitation contraction coupling. So we can see here in this way, these are the intercalated disc which is visible here. So intercalated disc are the part of cardiac muscle cell sarcolemma, and they contain gap junction and desmosomes. So we can see here like this, the intercalated disc which is showing the desmosomes, and the gap junction we can see the nucleus which is single. Cardiac muscle fiber is showing in this way in the gap junction along with the desmosomes and intercalated disc. Now cardiac syncytium is a network of cardiomyocytes connected by intercalated disc that enable the rapid transmission of electrical impulse through the network, and enabling the syncytium to act in coordinated contraction of the cardium, and there is an atrial syncytium and ventricular syncytium that are connected by the cardiac connection of the fibers. And electrical resistance to the intercalated disc is very low, thus allowing free diffusion of ions. In the case of ion movement along the cardiac muscle fiber axis is such that the action potential are able to travel from one cardiac muscle cell to the next and facing only slight resistance. Each syncytium obey the all or none law. In this photograph, we can see the transverse section of the cardiac muscle. The nucleus is located centrally, and individual muscle cells are surrounded by endomysium. So we can see endomysium, centrally located nucleus, endomysium with fibrocyte nuclei, then sarcoplasm with central nucleus, sarcoplasm with nuclear profile absent. Then endocardial lining, simple squamous epithelium, that is endothelium, subendothelial and connective tissue, modified heart muscle cell, and we can see also the modified heart muscle cell as a Purkinje fiber. Purkinje fiber inner wall of the heart is showing in this photograph. Now come to the next, that is the smooth muscle tissue, which is visible here as a spindle shaped cells with a nuclei, and the characteristic spindle shaped cells with central nuclei arranged closely to form the sheets, and no striations are there, and the function is to propel the substance along the internal passageway. They are involuntary control, and location which is found in mostly in the wall of the hollow organs. Smooth muscle is an involuntary non-striated muscle, so called because it has no sarcomere and therefore no striations. It is divided into two subgroups, single unit and multi-unit smooth muscle. Within single unit muscle, the whole muscle or bundle sheet of the smooth muscle cell contract as a syncytium, and smooth muscle is found in the wall of a hollow organ including the stomach, intestine, bladder, uterus, in the wall of passageway such as blood, lymph vessels, and in the tract of the respiratory, urinary, and reproductive system. In the eye, the ciliary muscle and the type of the smooth muscle is dilate and contract the iris and alter the shape of the lens. In the skin, the smooth muscle cells are, and those of the erector eye causes the hair to stand erect in response to the cold and the fear. In this photograph, we can see the smooth muscle tissue highlighting the inner circular layer. Nuclei then rest on the cell in pink, and outer longitudinal layer nuclei that rest on the cells and the serous membrane facing the lumen of the peritoneal cavity. So this is the outer, we can say that outer longitudinal. So this one is the outer longitudinal. In section it appears round, and this is the inner circular layer. That is why it appears like this. So this is a spindle shaped cell with the nucleus. We can see in this way like this. Now come to the next thing, that smooth muscle cells known as myocytes and are spindle shaped cells with this wide middle and tapering ends, and like striated muscle, and can tense and relax. And the relaxed state, each cell is 30 to 100 micrometer in length, and there are no myofibrils present, but much of the cytoplasm is taken up by the protein of myosin and actin, which together have the capability to contract. In this case, most smooth muscle is of single unit variety, that is either the whole muscle contract or the whole muscle relax. But there is a multi-unit smooth muscles in the trachea we can find, and the large elastic arteries, and in the iris of the eye also. That is a multi-unit. And single unit smooth muscle, however, is the most common and lines blood vessels except the large elastic arteries and urinary tract and the digestive tract. So we can see here that the body dense here and the attachment to the wall like this, and the uncontracted stage they appear like this, but in contracted stage they appear in this way. So this is the section of the smooth muscle in the appearing like this, which is the dark dotted structure is the nucleus of the cell. So this is all about the muscular tissue. Thanks.